Good morning. My name is Jennifer McNab. I'm an associate professor at Stanford University, and I'd like to thank the organizers for the invitation to be a part of this session. I'll be speaking today about comparing MRI microstructure maps and tissue clearing 3D histology in the same human brain specimens. The development of MRI methods for mapping properties of tissue microstructure continues to be an extremely active area of research. Instead of mapping non-specific diffusion tensor parameters, there is a strong focus on developing ways to access highly specific and biologically relevant features, such as axon diameters, myelination, neurite density and dispersion, and even a recent paper aimed at capturing some sensitivity to changes in glial morphology. Improvements in MRI hardware, such as stronger, faster gradients, are really helping this field evolve. More robust and efficient encodings enable a more diverse set of encodings to be acquired with a higher level of precision. And this provides a richer data set that can hopefully support more sophisticated models of tissue microstructure. Still, we are always relying on indirect measurements in MRI, and therefore multimodal comparisons are essential to evaluate whether we have successfully achieved the specific sensitivity to tissue features that we are aiming for. One approach to evaluating MRI microstructure maps is to perform MRI scans on postmortem tissue and then subsequently histology in the exact same tissue. In this way, it's possible to capture information about some of the histological components within an MRI voxel. Conventional histology usually involves sectioning the tissue and mounting very thin slices on slides. The tissue components of interest are identified using either a chromogenic or fluorescent label, and then the tissue slides are imaged with light microscopy. Unfortunately, some microstructural brain features are difficult to capture with 2D histology. For example, axonal fiber bundles and blood vessels project in 3D, and the curved nature of the cortex can make it difficult to obtain slices of tissue that accurately reflect the distinctive layering patterns. This recent review paper highlighted some of the difficulties associated with inferring on 3D microstructure from 2D samplings, showing convoluted structures, complex distributions of tissue components, and the possibility of failing to capture tissue components that are sparsely distributed. Still, it is possible to generate 3D volumes from 2D histology. This paper detailed some of the strategies to make this work and also highlighted some of the remaining challenges. Adjacent tissue slices can be distorted differently when placed on the slide, and here they demonstrate an approach to better align the slices. Still, they show some residual level of misregistration. For this reason, there's interest in performing histology on intact volumes of tissue. Tissue clearing aims to reduce the opacity of tissues such that optical imaging of intact tissue is possible. When combined with fluorescent labeling, it allows for whole brain 3D imaging with subcellular level specificity. Now, tissue clearing is a century-old approach that has been reinvented with recent developments in reagents and protocols, efficient fluorescent labeling, and rapid volumetric imaging by light sheet microscopy. Tissues are opaque because different components of cells have varying refractive indices, which cause a strong scattering of light. And so the tissue clearing process involves removing lipids, which are a primary source of this scattering, and also various solutions that enable refractive index matching. There are many, many different protocols for tissue clearing, but generally they all have these steps: fixation of the tissue, some sort of chemical treatment, removal of the lipids, labeling, refractive index matching, and microscopy. I'm going to show some results using the Cleari protocol first, and this involves embedding a hydrogel in the tissue, which helps to lock the proteins and nucleic acids in place. And the lipids are then washed out using a detergent. There's a stain of the proteins. Proteins or nucleic acids are labeled with fluorescent markers, and imaging is done using light sheet microscopy. Now, these images are very exciting. We can see a whole mouse brain here, which is about the volumetric limit that we've been operating at. Here we can see a whole mouse brain with exquisite visualization of individual neurons. We also seen some very nice results in larger tissue samples of a rhesus macaque, showing some of the expected patterns in the visual cortex. And again, here with the very easy to recognize Gennari stripe. The original Cleari paper did demonstrate the technique in human tissue, although there are some notable differences when working with human tissue. Human tissue tends to be even more heavily myelinated, and therefore if passive clearing with detergents is used, it can take a very long time. Further, it's not possible to rely on transgenic labels, and many of the antibodies needed to visualize features of interest are quite large, and it's non-trivial to have them propagate evenly throughout the tissue cuboid. Still, Nicholas Weisskopf's group demonstrated the ability to use a number of different types of stains in cleared human brain tissue, and also the ability to stain fairly evenly over several millimeters of depth. When comparing MRI with tissue clearing data, the MRI must be performed first because the elimination of the lipids and perhaps other aspects of the tissue processing pipeline do eliminate most types of MRI contrast. When cutting the tissue, we found it essential to perform MRI acquisitions and photography at each stage of the cutting, such that the frame of reference relative to the whole brain is not lost. Now, imaging fixed postmortem tissue does involve a number of important considerations. It's been shown that the postmortem interval, the time between death and fixation, is best to be kept short. This is showing that the axal diffusivity does get longer as that postmortem interval is longer, gets longer. Fixation is achieved with immersion fixation for human samples as opposed to perfusion fixation with animal samples, and that means it'll take at least 6 to 8 weeks to usually fix all the way through a whole brain, or that can happen over a much shorter time frame if you're just using a much small section of tissue. T1s and T2s are stable in the fixed once the tissue is fixed, but they tend to be much shorter than compared to in vivo, and proton density is lower. It's also been shown that the T1 and T2 weighted contrast between white matter and gray matter changes, in fact, inverts in some cases. And so that's a consideration for any structural imaging. And also because you're imaging at room temperature, you have a much slower diffusion rate, means you require more diffusion weighting to get the same level of contrast. In terms of an experimental setup, usually the brain is surrounded with some sort of non-protonated fluid, a perfluorocarbon. The idea is to have a susceptibility match surrounding the tissue, but also not to have too much signal from the fluid surrounding the tissue. There's a desire to eliminate air bubbles. Sometimes we put it in a vacuum overnight to do that. Also because of the susceptibility difference with air pockets. And you want to pack it in a way so that it's physically stable in the scanner, but also not squished so much that it deforms the shape of the general shape of the brain. It's important to keep it temperature stable because this can impact our diffusion and our T1 and T2 measurements. Running the scanner for long periods of time can sometimes heat up the bore. You also ideally would like a nice tightly coupled RF receive coil, and the MGH group showed a nice example of this recently. There's also the consideration of the availability and cost of potentially several days of scanning if you want to do high resolution diffusion imaging. The shorter T2s and the stronger diffusion weighted needed means it really does take quite a long period of time to get these types of data. And then there's also issues just in terms of keeping the scanner running continuously. These scanners aren't set up to do so, and there can be lots of pop-up warnings, and you can run the risk of gradient overheating from running it continuously for so long. There's pesky SAR monitors, which aren't really relevant to the postmortem tissue, but can be difficult to turn off. And remote monitoring can be really, really helpful to run these types of experiments. In terms of acquisition strategies, there's no motion, and there's some changes in T1 and T2 to consider. The fact that T1 and T2 changes have meant that balanced SFP, which has this funny T1 over T2 contrast, provides really good gray-white matter contrast. And so balanced SFP with some phase-like cycling has shown to work really well to get really high-res, beautiful structural images in postmortem brains. For diffusion imaging, there's no motion, so you can use these segmented acquisitions to get really high-resolution, low-distortion images. And you do need B values about 3 times higher than you would use in vivo to get a similar level of contrast. Still, if you take these things into consideration, you can get absolutely drop-dead gorgeous, beautiful data that has a lot of value on its own, even before you compare it to any histology. Now, in terms of the types of stains we looked at with the Cleari data, we focused on a small molecule stain, the propidium iodide, which stains the DNA and RNA and is fairly easy to use. We also used a neurofilament stain, which is requires a much larger antibody, but provides a nice visualization of some of the fiber structure. And we wanted to look at that because, to start with, just because we felt like we have a fairly good idea of how to map fiber orientations with diffusion imaging. And that was a good place to start with these comparisons. A really big part of these comparisons is getting an accurate alignment between the MRI and the optical images. We found that using the vessels as landmarks, both for guiding the registration and evaluating the accuracy of it, was incredibly useful and gave us a lot of assurance that there was a close correspondence between the 2 types of images, and we could do a very detailed voxel-to-voxel comparison. There's also a need to extract, in some sort of quantitative way, the parameters, histological features from the histology data. Luckily, there's pretty well-defined ways to do this for fiber structures. There's the structure tensor analysis, which we were able to do in 3D with this data, and relies on image intensity gradients to identify the boundaries of fibers. We also spent some time looking at the volume over which you look at these image intensity gradients and how that relates to the resolution of your microscopy data and how that affects the ultimate fiber orientation distribution that you estimate. And it turns out it is quite important to think about that closely and think about the size of the structure within the histology that you're trying to capture, and how that relates to image resolution and how that might relate to your MRI data and the sensitivity you have to different sizes of features in the MRI data. This is an example side by side of the Cleari neurofilament stain with the structure tensor estimates at each location of each MRI voxel, and then a diffusion tensor estimate of the principal diffusion orientation at each of the MRI voxels. And the color map is showing the angular difference between those 2 orientation estimates, showing in the blue, very good agreement, low angular difference. Some disagreement around the edges of the sample, but generally a pretty close correspondence. We also looked at a comparison of diffusion tensor orientation estimates and orientation estimates in the Cleari using structure tensor, but on the vascular, the Cleari data that shows the vasculature rather than the neurofilaments. And this was interesting because there's been some open questions about whether the vasculature contributes at all to some of the white matter orientation estimates that we get with diffusion. And in this case, we didn't see any correspondence between the 2. It is interesting though, because it has been shown that the vasculature does tend to run along similar orientations to a lot of the white matter tracks. And there's some open questions about how this might impact some fMRI measures as well. We've also experimented a little bit with some iDisco in human tissue samples, which is a different type of tissue clearing protocol. The lipids are generally dissolved rather than washed out with a detergent. And this seems to work pretty well. And we've had some success looking at melanin concentrating hormone, and also myelin basic protein and DAPI and neurofilament stains. It seems like another promising approach for doing these types of comparisons, particularly in human tissue. This is lastly, I just want to highlight this really nice study led by my collaborator, Casey Halpern. He was very interested in looking at the connectivity of the nucleus accumbens. Dysregulation of the nucleus accumbens is implicated in a number of neuropsychiatric disorders, and there's an interest to treat this area directly with deep brain stimulation, but the initial results have been kind of variable. And one hypothesis is that the nucleus accumbens is quite functionally heterogeneous, owing to different parts of the nucleus accumbens connecting with different parts of the brain. Casey was very interested in us to look at this with tractography and see if we could segment different parts of the nucleus accumbens based on different tractography evidence of different connections with different parts of the brain. And then here is also an example of using Cleari to try to highlight some of these different connections and the orientations of some of the pathways entering and exiting the nucleus accumbens. And also a very nice comparison with some of the tractography results in the same sample. And then this is in vivo data where we did do a tractography-based segmentation based on differing connectivity profiles of different parts of the nucleus accumbens, which seemed to line up quite well with some of the known histology. And what was really beautiful was another additional affirmation of this segmentation was some results from a deep brain stimulation that was performed. And you can see retrospectively here, we aligned the tractography-based segmentation to show whether the electrodes were in the two different tractography-defined parts of the nucleus accumbens or in the same tractography-defined section. And you can see on the left side, on the top image, there's two electrodes that are in different parts of the tractography-defined sections of the nucleus accumbens. And in that case, they saw a very differential response when they stimulated from each of those two electrodes. Whereas on the bottom, when they appear to both be in the more inferior portion, they showed a much more similar response. So it's a really nice sort of multimodal study to try to answer a very specific hypothesis. So in summary, MRI capabilities for mapping biologically relevant parameters continue to evolve. Direct comparisons with advanced histology, such as tissue clearing 3D histology, provide new opportunities to improve the interpretability of MRI biomarkers. I want to thank my group members and my funding sources and the organizers and participants here again for the opportunity to participate.